Hi, I'm Aiden Smith and I'm here to discuss passive and active transport. Passive transport is when substances are moved across the lipid bilayer without the use of ATP, and active transport is with use of ATP. I'm going to start out with passive transport. There are 5 kinds of passive transport. The first kind is simple diffusion. Simple diffusion is when nonpolar and lipid-soluble substances such as oxygen and carbon dioxide are diffused through the lipid bilayer. In facilitated diffusion, there are 2 different kinds of that. Carrier-mediated facilitated diffusion, and the carriers are transmembrane proteins, and they envelop large solutes that are too large to pass through the lipid bilayer, such as sugars and amino acids, and they're limited by the number of receptors, and when all the receptors are full, the cells are said to be saturated. In channel-mediated facilitated diffusion, lipid solubles are moved through aqueous channels through the membrane. They're selective due to pore size and the charges of amino acids on the inside of the carrier. The final type of passive transport is osmosis. Osmosis is when water diffuses directly through the lipid bilayer or is transferred through in specific channels. Next is active transport. There are also 5 kinds of active transport. The first kind is primary active transport, and that is used in a sodium-potassium. Sodium-potassium pump works as a pump protein is on the top of the lipid bilayer. And sodium binds with the pump protein, causing the phosphorylation of ATP. And that causes the pump protein to switch, which shows here is opposite. And that causes the sodium to be re-released back into the extracellular fluid. And that, in turn, causes potassium ions to bind to the pump protein. And that causes the phosphate group that is in the pump protein to be released inside the cell. The loss of the phosphate restores the original conformation of the pump protein, and it is ready to bind again and repeat the cycle. The second type of active transport is secondary active transport. It also uses a sodium-potassium pump, but sodium is pushed outside the cell against this concentration gradient and it stores up energy. And as the sodium floats back into the cell, with the help of a carrier protein, it pulls along with it glucose, thus ending secondary active transport. The third type is exocytosis, and this is when a cell gets rid of a substance. The substance that is in the cell is contained in a vesicle, and the vesicle migrates to the plasma membrane. V snares for vesicle snares and T snares for target snares bind together at the plasma membrane and corkscrew together, thus releasing the substance out of the cell. And then the final stages of active transport are clathrin-mediated endocytosis and receptor-mediated endocytosis. In clathrin-mediated endocytosis, a substance that is being taken into the cell is enveloped in a clathrin-coated pit, and it's called clathrin-coated because of the bristle-like structures on the inside that coat the whole cell. That pit is enveloped and detaches from the plasma membrane and migrates into the cell. There, it binds with an endosome and is dealt with in one of 3 ways. The first way is it is recycled back to the plasma membrane and used there. Or it binds with the lysosome and is digested and used by the cell. And the third way, it's released by endocytosis on the opposite side of the cell. In receptor-mediated endocytosis, it works pretty much the same way, except for the substance has to bind with certain receptors. And the receptors on the outside of the clathrin-coated pit are very selective. They only bind with certain substances. And when those are full, it envelops the substance and migrates inside the cell, and is dealt with in also one of the 3 ways. It's either recycled to the plasma membrane, binding with a lysosome and used by the cell, or gotten rid of by endocytosis. And that is our discussion on passive and active transport.